Hello, Katarina. Hello, Milka. So, thank you very much for your invitation and for organizing such an important event, which, as you said today, is absolutely necessary, and we need to do this as often as possible. I'm very happy to see you. I will now let you give us your presentation, and if there's any questions, we'll talk again at the end of your presentation. You can now start sharing your screen. Here it is. I will now switch off and see you a bit later. Bye, thank you. A very good evening to you all. As Dr. Milka Popović has already said, I'm a speech therapist and I've got a PhD in biology. I work in Dr. Selaković surgery, dealing with psychiatry and diagnostics of early developmental disorders. And today I'll be telling you about a small part of a large research which was conducted here in Serbia within the Biological Research Institute Siniša Stanković and the Institute for Molecular Genetics and Genetic Engineering. The results that I'll be telling you about today refer to the research of specificities of gut microbiota in children with neurodevelopmental disorders. So very briefly, I'll remind you that neurodevelopmental disorders are disorders which appear in early childhood, usually before the age of 3, and they can cover one or more developmental spheres. So we're talking about speech and language disorder, developmental dysphagia, specific developmental disorders of motor functions, attention deficit and hyperactivity disorder, ADHD, mixed specific developmental disorders, and the most complex form of these disorders is the autism spectrum disorder. So as you've heard a number of times already at this conference, the causes of neurodevelopmental disorders are not known. The largest number of research looked at the etiological basis of autism as the heaviest form of developmental disorders. And the greatest number of these studies studied the genetic origin of autism. So the results of these research showed the importance of genetic factors, genetic variants found in the population, with the presumption that such genetic variations, with their small to medium effects and specific combinations within an individual genome, and sometimes in the interaction with the central factor, can lead to the clinical autism. Some other studies showed the importance of genes which code or regulate the expression of genes used in the creation of synaptic functions, or rather point to the importance of these rare monogenetic forms of autism. However, around 90% of the cases of autism is idiopathic, meaning that the etiological basis is unknown. It's therefore presumed that this sensitive genetic basis is influenced by environmental factors as triggers. The special attention in literature over the last few years has been given to research of comorbidities, mainly, I say, autism as the most difficult phase of developmental disorders, which has been studied the most. As one of the most frequent comorbidity are listed allergies, respiratory infections, sleep disorders, neurological disorders such as epilepsy, and literature will also tell you that up to 40% of autistic patients has a certain form of epilepsy, but that the number of patients where the subclinical epileptiformic changes are present without clear clinical correlation are much higher. But special attention is given to gastrointestinal disturbances as a comorbidity of autism. So it is believed, and the results of certain studies have shown that gastrointestinal disturbances such as diarrhea, constipation, bloating, and gastroesophageal reflux are much more frequent in people with autism and other forms of neurodevelopmental disorders. The literature will give you information that diarrhea and colic disturbances are 7 times more present in people with autism than typically developing children. It is therefore presumed that these gastrointestinal disturbances could contribute to the appearance of certain other symptoms typical of autism, such as sleep disorders, sensory dysfunction, and behavioral changes such as aggressive, impulsive, and destructive behavior. It is also presumed that these gastrointestinal disturbances are a consequence of changes to the gut microbiota, whether it's manifested with a disturbed relation between the good bacteria, the bacteria which are a part of the intestinal microbiota in grown-ups, or in the overgrowth of potentially pathogenic bacteria. Very briefly, I'll remind you that intestinal microbiota is a complex environment of microorganisms living in symbiosis with the host and contribute to the health of the host. Their roles are numerous and include protection from pathogens, induction and modulation of immune response, digestion of nutrients and medicines, synthesis of vitamins and amino acids, and especially lately the role that they play in development and functioning of the central nervous system, which the microbiota conducts through modulation and expression of genes in many organs. And also, when it comes to the central nervous system, then the limitation of availability of precursors of serotonin, tryptophan, as well as using certain metabolites such as fatty acids, short-chain fatty acids. The literature contains data that children with autism have a different microbiota compared to children, typically developing children, in the overgrowth of some pathogens, especially those belonging to Clostridia clusters, or in the reduction of bacteria which are a part of the healthy microbiota, such as bifidobacteria, and an increase in lactobacilli. Now, bearing in mind all this information, as well as information from clinical practice, which point to the fact that gastrointestinal disturbances are also present among children with other types of neurodevelopmental disorder, not only in autism, and also bearing in mind the fact that the mechanisms of forming certain other developmental disorders are also largely unknown, and that there is very little research of intestinal microbiota. Under this research, we have not defined the following. We have defined the following goals, which was to test whether the diversity of gut microbiota is different between children, typically developing children, and those with neurological disorders, and those types of bacteria which, to a greater or lesser extent, were statistically different between NDD patients and the control group. What I feel I need to point out is that according to our knowledge, this was the first study of intestinal microbiota performed on children from a wider NDD spectrum, and that all the research, according to our knowledge, was conducted in children with autism. Now, the research covered 36 NDD patients with a diagnosis of neurodevelopmental disorder and 28 children typically developing children. Now, what is important we need to point out is that when collecting samples in the control group and in patients was a long process and not at all easy, because the most important criteria for selecting patients was that the children should, before 3 months before giving the feces sample, were not submitted to any nutritional or medical treatment which could have influence on the intestinal microbiota and its composition. So that means that 3 months before giving samples, the patients had not taken probiotics, prebiotics, antibiotics, or other supplements, or any sort of anything that could impact the intestinal microbiota. This was, of course, very difficult because for the simple reason that children with neurodevelopmental disorders, mainly with autism, often have respiratory infections, especially those which are more complex, with the middle ear infection, which are treated with oral application of antibiotics. So it's very difficult to collect samples using the same criteria. And so using the same criteria, we selected members of the control group, but there we had an additional criterion of not having any gastrointestinal disturbances or any sort of developmental retardation or blood relations with the patients. Now, I'll tell you about the samples very quickly now. Now, in this image, you can see the diversity of intestinal microbiota. Every spot here refers to one genetic amplicone, a certain strand on the DGG gallum. The comparison of fecal microbiota of our patients and the control group was conducted using the DGG analysis of genetic amplicoma, obtained using 3 sets of primers specific for lactobacterial primers, bifidobacteria primers, and universal primers. So, as I said, each point here corresponds to one DNA amplicone, or rather to one strand on the DGG graph. So those amplicones which were statistically different, or their presence was different in the group, between the patients and the control group, have been isolated, cloned, and sequenced. Here we see the identification of these bacteria obtained by sequencing the chosen amplicona. Now, on this slide, you can see the comparison of the diversity of intestinal microbiota between a whole group of patients and the control group on the left-hand side. Statistically, significant difference in the diversity of intestinal microbiota was obtained through using the DGG profile analysis, obtained using the universal primers, using LB, lactobacillus specific primers, and also using the analysis of the primers obtained by bifidospecific primers. It is very clear that the diversity of intestinal microbiota is largely smaller than the diversity when it comes to intestinal enterobiota of the control group. So when comparing individual groups of patients with the control group, statistically important differences in the diversity of intestinal microbiota was obtained when analyzing the DGG profile from lactobacillus-specific primers, and this trend was also noticed using the analysis of DGG profile obtained by bifidospecific primers. On this table here, you can see the bacterial types of bacteria that have been identified using the sequencing those DNA amplicons, which are statistically significant between the control group and the group of patients. Our results showed that the microbiota in patients is significantly different from the microbiota of members of the control group, primarily in view of the overpresence of bacteria, of types of bacteria which are considered potentially pathogenous and which here have shown a high level of relation from the Clostridia strain. So the Bartlett strand, which before the classification was known as the Clostridium Bartlett, was significantly much more present in the people with mixed disorders compared to the control group. Also, a higher presence was noted in other presence, but statistical significance there has not been obtained. The glutaidium strand, DSM strand, was statistically more present in the group of patients with developmental dysphagia compared to the control group. Now, in literature, you will find information which presume that these organisms, such as glutaidium, can contribute to the disturbance in the metabolism of sulfates, which is often noticed in patients with autism and which is manifested with a lower level of sulfates in the blood, higher extraction in the urine, disturbance in methylation process, and oxalate stress, all conditions which have also been brought in connection with autism. But it is presumed that the overuse of oral antibiotics, which, as we can see in clinical practices and also data from literature, is very frequent among children with autism, and it contributes to it disturbs the normal intestinal microbiota and the creation of intestinal dysbiosis and allowing an overgrowth of these potentially pathogenous bacteria. And it is therefore presumed and believed that this way there is an overpresence of their metabolites which can be neurotoxic, such as paracrasonphenol internal compounds, which in literature have already been proved that they can lead to anxiousness. The hypothesis of Van Bolton says that a form of autism known as regressive autism, where the child has normal development until the age of 3 and then they lose acquired skills, that this could be the a result or a consequence of this disturbed situation in the intestinal microbiota due to the overuse of antibiotics and the growth of pathogenous bacteria from the Clostridia strain, which are resistant to antibiotics. The results of our research showed that in patients with neurodevelopmental disorders, there is a presence of a lower presence of lactobacteria, such as Faecalis, Lactobacillus rhamnosus, Dialistres inviscus. We can see the Lactobacillus rhamnosus is statistically less present in the group of patients with neurodevelopmental disorders compared to the control group. Now, Dialestrin inviscus was also statistically less present in the group of patients compared to the control group. In literature, you can find also information that Lactobacillus rhamnosus or the continued use of Lactobacillus rhamnosus can lead to the changes in the stress response in the through the reduction of cortisol, and that it can lead to depressive or anxious behavior through the regulating the certain receptors in the brain. Our results have also shown that certain bacteria which produce certain acids are less present in the group of patients than in the control group.
Butyric acid is one of the fatty acids. Butyric acid is one of the fatty acids that it is not important, and it produces this connection, and it develops to the balance of the hematocephalus and gut barrier. It is also well known that in the literature you can find data that the increase of the lack of barrier is oftenly described with patients of autism spectrum. Bacteria that are strong contributors to the deuterium acid are Botulinum pilicocorum, F. prausnitzii, E. rectale. R. champagnolysis, although it is not a direct compound or a cause of the butyric acid, by not digesting the material, it provides the material and energy for the other beneficial bacteria, therefore bringing to the benefit of the gut microbia. F. prausnitzii is one of the more dominant species in gut microbiota, and the lack of this bacteria are seen in the patients of irritable colon. Our results have also shown that patients with generic or more general problems have less and lack of lipidobacteria as opposed to control study group. As much as 12% of the sample study has noticed the lack or not existence of lipidobacteria, and the other ones are not even detected in the sample size. Lipidobacteria are the first ones that show in the colon and present an important part of the gut of children, but it's also a part of gut microbiota in persons, in adult persons. It is very important to have it in order to regulate the immune response, to help B vitamins, and it can be suggested that some of the joint states comorbidity actually with patients who have problems with digestions, common infections, lack of B vitamins can also be connected to the lack of these lipidobacteria. And the literature also states that lipidobacteria can also be quite important and useful while treating some psychiatric diseases such as depression, anxiety, and anxiety. Namely, experiments that have been done on mice that have been chronically exposed to stress showed that treatment with lipidic phases have shown improvement on the axis, and it has increased the level of cortisol in blood. Other research that have been conducted on healthy mice have shown that this bacteria has decreased depressive moods. With lipidolongum, it has shown that it has antiseptic part on the aseptic role with mice, and that this doesn't exist in mice where there is lack of vagus, which shows the ways in which microbiota can infect the central system. Some other systems have shown that the treatment with lipidotrongylism can lead to decrease of anxiety level with mice. The thing that is in common for most of these studies is that they indicate the importance of the temporal and the importance of the real-time connection of the gut, which then shows the pre-adult period, which indicates the pre-adult period, and that only by coagulating specific bacterial species can have a positive effect, and that this can be shown in the less amount if it is done in the adult period. This is coinciding again with another clinical studies where there is notice a comorbidity of these conditions, in this case gastrointestinal problems and their treatments. The reproduction can be improved, and the reduction, even in part, can be brought by doing this. within this research, we wanted to see how much the gut microbiota of each special studies of each patient is homogeneous. When we analyze DGG profiles obtained by Latin bacillus and BB-specific primers in all 3 sets of primers, the most common homogeneity of groups is seen with control studies. The least homogenic level is seen in the receptive problems with language disorders, so excessive receptive language disorders group. Then we wanted to see to what level the microbiosis of the individual levels is different from one another, therefore, or conversely, how similar it is. And when we analyze the DGG profiles acquired by lactobacillus-specific primers, the highest level of similarity has been seen in gut microbiotes in the patients with mixed development and as well with PDD. When we analyze PGG from BB-specific primers, most similarity has been seen with patients that have mixed developmental disorder and child autism, when all 3 sets of primers, that is DGG profiles from all set of primers, a high level of similarity has been seen in gut microbiotas in children with mixed developmental disorder and pervasive developmental disorder, and the ones with children autism, and mixed developmental disorder with children autism. This has been a very significant result because it has shown that the gut microbiota is clinically similar to the level of the deformities with development, and there are many cases with difficulty of these development. We want to say that the gut microbiota is similar between these deficiencies and problems, so that indicates that gut microbiota can have a significant role in pathogeneity of these developmental disorders. When we also compared how much gut microbiota of each of these individual disabilities is similar to the control samples, when we analyzed DGG profiles acquired from all 3 sets of primers and acquired from lactobacillus-specific primers and BB-specific primers, we have seen that the least level of similarity with the control study has been among the patients with expressive receptive language disorder. In addition to this result has been quite significant because it has shown that the correction of the system of the less development disorders, such as disorder of expressive receptive language, can be significant when it comes to rehabilitation, which is somewhat in accordance with the clinical practice where if we can see the problem in real time and on time with gut microbiota, we can actually see some symptoms in advance that among the patients with this diagnosis, and therefore it can lead to faster rehabilitation. On the basis of all of these results, we can conclude that gut microbiota of the patient with a wide spectrum of NDD can be different from gut microbiota of typically developed children. A lower diversity has been noticed of bacteria that are part of the gut microbiota of adults, such as lactobacillus and bifidobacteria, and increased incidence of several potential harmful bacteria are also shown, especially the ones that are closely related to the ones that are related to Clostridium species. Our results can provide a cause for further studies of gut microbiota dysbiosis in NDD patients, and it can also elucidate possible pathogenic, possible mechanism that this bacteria can use in order to how this bacteria can actually influence the patient. According to these results, it can be concluded that disturbances in gut microbiota can have an impact not only on the occurrence of gastrointestinal symptoms in children with NDD, but also on the occurrence of some clinical symptoms. According to these studies and data that can be found in literature, we can assume that early screening of gut microbiota can be potentially useful for prevention of gastrointestinal disturbances, but also to follow changes of behavior of people with autism and NDD. Last but not least, our results lead us to conclude that the composition of gut microbiota and supplementation of chosen strains, promoting strains that are found to be deficient in those patients can be an additional therapy while treating people with NDD and gastrointestinal disturbances, which are often comorbidities. I appreciate for your attention. Hello, hi, can you hear me? Thank you for this wonderful presentation. It is great to see and to hear all the novelties that you have discovered within your project. The thing that I would like to ask you, if you don't mind, is related to the group of children that has been tested, which amounts to 36. Did all of the children have gastrointestinal disturbances? 57% of these children had gastrointestinal problems and disturbances. So it doesn't have to mean that the child from with SAD has a disturbed gut flora as opposed to children who are typically developed. So they can have different gut flora and not have gastrointestinal need. So it could be quite interesting to see, actually, to screen microbiota of these children, because when parents arrive and when we ask them if children have gastrointestinal problems, they often say that they don't have or they don't know. Like, sorry to interrupt you, but I would like to add that it's quite difficult when you interview children with parents. It's quite difficult because you need to ask them very specific questions. When you ask parents if they have gastrointestinal problems, parents usually respond that they do not have, and then then we ask them if children have regular stools, have they ever had problems with diarrhea, and then when you specifically ask them for specific questions, then you can actually gather some data that can help you to present some objective data about gastrointestinal problems. Yes, I understand. However, I have had experience after that all of these questions and everything is okay, and after we have done the stool sample, that we can see that there are some problems. Yes, indeed. However, these are some clinic aspects that we need to follow, and regarding this, then we send the child to specific analysis, which you talked about today. I understand. Now, the application of specific strains of bacteria, regarding that point, in the market currently we do not have that opportunity. Now, wouldn't this be great that we had this opportunity? Yes, it would be great to have this. However, now through feeding effect, we need to, such as using some bacteria, we can use this help in order to give the body some kind of help to feed and reduce any kinds of intestinal problems. This is related to the studies which I read for today's speech. Therefore, using these kind of pills, they can actually increase the level of bacteria. Now, it would be ideal to have any kind of sorts of help that would aid in this. Nevertheless, now we need to rely on feeding in order to regain any kind of balance in gut microbiota. Now, this is one of the first research in our country. However, there are numerous that talk about gut microbiota and the way that it affects on the central nervous system. This is definitely one of the things that a person who is diagnosing the problem should not overlook. Yes, of course. Now I'd like to add that the simplest intervention is the regular diet, because this is the diet for good microbiota. Now, commonly among the children, we can see that they do not like this kind of food that actually diminish this kind of bacteria. So this is a vicious cycle. Yes, I would agree. This is why it is important to apply multidisciplinary approach. So it's important to have expertise from different kinds of areas. So this is not from this kind of... Now, today's clinical pictures are very much complicated than the ones that used to be before. Now, today, there isn't clinical dysphagia that is clear. There is always something combined. It is quite important to have a multidisciplinary approach where all the aspects of the so-called one organism, one child, will be encompassed and will be treated. Only in this way will we be able to treat in the maximum way, as opposed to the ones that children usually get. Thank you so much. I would agree completely. Thank you for thank you for sharing this wonderful presentation. Thank you for calling me. Goodbye.